Hi, everybody. I hope you can all hear me okay. I guess we'll get started, but before we do that, I do want to go over a few things. So I don't know if you guys saw this on Canvas, but I posted a step-by-step on how to solve for PI for the different types of amino acids. If you guys are still struggling with that, you can look at that and it should help. Another thing I've been getting a lot of emails about is the practice questions for Dr. Ferreira for her second lecture, the proteins and peptides one, I believe, on how to solve for those. So if you guys still need help with that, I can go over it at the end of this review session, and you guys are welcome to stay for that. Just remind me, of course, if you need it. And then as for last week's review session, we had a kind of confusion with the anions and the cations. So I did rewatch her video or her lecture, and she did mention that she was talking about gel electrophoresis, in which case the anion would be positive and the cation would be negative. But that's not really that important. You don't have to know anything about gel electrophoresis for that because she's not going to ask you about it. That's too chemistry. And she was just using that slide to introduce the concept that amino acids can change their charge depending on whether they're in acidic or in a basic solution. Okay, so is anyone still confused about the Q&A for tomorrow? Because I've gotten a few emails about that as well. Because if not, I can just like go on to the next thing and we could start, or I can re-explain.
Oh, I have a quick question. So regarding tomorrow, will we be able to see the questions after we take the quiz?
I think what's going to happen is if you submit it, it's going to only show like your score, like whether you got like a 42 out of 48 or whatever it is. And then at 2 p.m. when you enter the Teams meeting for that, we'll change the settings and then you'll be able to see like which ones you got wrong in all the questions, and we'll go over it.
Thank you.
You're welcome. Are there any other questions about that?
Yeah, I have a question.
Yeah.
What is the Q&A tomorrow? I don't really understand. I didn't hear anything about it.
Oh, okay. Yeah, I'll just re-explain it then. So tomorrow, I think at 11:15 on Canvas, you're going to have access to a practice test. It's going to be about 48 questions, and it's going to be 12 questions per course. And it's going to go over the last 2 weeks of material, I believe. And then you're going to have like about a 2 or 3 hour window to take that. So I think it closes at 1:45 p.m. tomorrow. So once you start the quiz, make sure you have like 70 minutes to complete it because it's timed. You might not need that long, but if you need that long, make sure you have at least 70 minutes because it's going to close at 1:45. And then at 2 p.m., you enter the Teams meeting for the Q&A sessions, which I think you guys all got invited to on Teams. And then we'll go over, all the TAs are going to be there. So we're going to go over the answers like one by one. And then you guys are welcome to ask us any questions that you may have about anything. So does that answer your question?
Yes. So it's just like a tool for us to use to study, right?
Right, exactly. It's just a practice test. It's not going to into your grade or anything. It's just for fun, not for fun, but like for practice.
Thank you so much.
You're welcome.
Is there a practice test can like be up all weekend?
I believe we'll let you have access to it for, until forever, I guess, until we're done with this semester. I think so, but I'll have to double check. We'll probably post a Canvas announcement with all the details, but I'm pretty sure you'll be able to like look at it again like later on in this block.
Okay, perfect. Thanks.
You're welcome. Any other questions?
This will simulate our typical testing environment, like we have to take it with Proctorio. Is that correct?
No, it's not through Proctorio. It's just like a regular Canvas quiz, but it will look exactly like the test does. It just doesn't have like the camera and everything that Proctorio has.
What time does the quiz open again?
I think it's at 11:15 a.m. tomorrow. We'll probably post a Canvas announcement about it later today. So yeah, just look for that and it'll give you all the extra details. Are there any other questions about that? No? Okay. All right. I think that's everything I wanted to go over with you guys. So I could just start lecturing. The only thing I changed about my review session today is that instead of having practice questions throughout the PowerPoint, I've decided to just make a Kahoot at the end that we could all play. You don't have to stay for the Kahoot because I'll post it regardless. But it helps to like do it like right after the review session, and we could all play together if that works for everybody. But if you have to leave, like you could just play it another time or by yourself later because I'm going to post it. Alrighty, so let's get started. Okay, so this lecture was relatively straightforward, I believe. So she just wanted to introduce collagen as a type of fibrous protein. And then she also gives you these examples. She has them bolded, so I memorized them last year. But I think it's like pretty straightforward, you know, collagen, you think keratin, which is in your hair and nails. And then tropomyosin may not be so obvious, but that's what is associated with actin in your muscle. So just memorize these bolded terms and associate them with fibrous proteins. It's important to note that collagen has mostly secondary structure, so alpha helices. It's rod-like, it's long, and it is not really soluble in water. Also note that it's more for structure rather than dynamic. So like while most globular proteins have like a functional role, like enzymes, like they do something, this is mostly just to help with the structure of your tissues. So know that collagen is found in your extracellular matrix. So your extracellular matrix is just the area around your cells that help form the structure of the tissue. And then also you find it in connective tissue, which is, you know, the area behind your epithelium or below your epithelium, I should say. And that just helps, you know, also form tissue's structure. I think the most important aspect of collagen is to know that it adds strength to a tissue. As for these, I numbered them from like first to seventh, but I think the most important thing to remember is that it mostly makes up skin and that it maybe remember this percentage. I highlighted it because I think I memorized it last year, but I think that might be too much information. I would, however, memorize that it makes up about 1/4 of, your 1/4 of all your protein in your body is collagen, because with collagen, you're going to remember 1/4 for a couple of things. So this is a good number to remember. We'll get back to this because she has a copy of this slide, and I'll tell you what you need to know from that. So last year, our TA told us to memorize the examples for each of these. So I did, but it shouldn't be too difficult. You just associate alpha helix with hair, feathers, and nails of alpha keratin. Super simple. For the beta conformation, this is probably lower yield because you don't find silk in humans, but it might be something to memorize. But probably the most important thing to remember about this is that collagen gives tissue its high tensile strength because it is very strong. And we'll go over what you need to memorize for the collagens because they're different depending on the type of collagen. Okay, so this is much more important than what we just went over. So remember when we learned about alpha helices, you know that it makes a right-hand turn. So collagen is kind of the exception to this rule. Its alpha helix makes a left-handed turn. So make sure to note the difference between the regular alpha helices that you find in most proteins and alpha collagen. So left versus right. However, when you make the triple helix, so 3 of these single alpha helices will come together to make a triple helix, which all together makes a right-hand turn. So make sure to understand that, and I kind of put a little table over here. I would memorize this sequence and know that these two are common sequences as well, where you have a glycine, then some amino acid, and then you have proline or hydroxyproline. But I would mostly just memorize that glycine is always in the 3rd position. So it's going to be every 3rd amino acid. That's the most important thing to remember here. Also know that the reason glycine is in every third position is that the third amino acid is going to have its R group facing into the alpha helix, and we know that the alpha helix has a small space in the center, so you need to have the smallest amino acid side chain that you can fit, which is glycine, because that's just a hydrogen. I also made a note to remember that collagen has 3 residues, so 3 amino acids per turn, and the alpha helix, in contrast, has 3.6 residues per turn.
Phoebe,
Yeah.
I have a quick question on that slide. Actually, can you go back
Yeah.
one?
Yeah, sure.
When you say third residue, it's I'm sorry. Can you go back one slide? Yes, right there. It's listing glycine x proline. So isn't that in the order of like N to C terminus? So glycine would be in the first position, but I noticed you said third position.
Okay, so what she said third position, and I know that's super confusing, but I think the point she's trying to make is that it's every 3 positions. Like,
Okay.
yeah, it's just going to be every third amino acid. So hopefully that clarifies it.
That does. So that'll be every, third amino acid will be a glycine, and that will be internal R group because it fits. That's
Right,
all we
exactly.
need to know. Okay.
Exactly. Yes.
Thank you.
You're welcome. Okay. So this is like a common repeating sequence that you're going to see. It's not going to be like the whole thing, but you're going to see it kind of frequently. And I think she shows an example of that in the next slide. But kind of be familiar with this sequence as being somewhat common in collagen. Yeah, and then knowing that it's a triple helix, like we already said, and then the triple helix makes a right-hand turn. So this is pretty much just a repeat of what we went over. She did want to emphasize that proline is probably the second most important amino acid in collagen because of the, it's just found in collagen pretty frequently. You're going to see that the large R chain of the proline is going to add steric hindrance, and this kind of causes the whole chain to stretch, and as a result, it winds up tightening the twist between the 3 alpha helices. So it's a lot tighter together. So that's all I would remember from that slide. Okay. And here she's just showing you kind of like if you're looking through the collagen triple helix, you'll see that there are hydrogen bonds, which I believe are between the hydrogens on the glycine groups. And so you have to know that the triple helix is held together by hydrogen bonds. Is there a question? I think somebody's mic is on. Okay. Oh, there's a question in the chat. So each singular helix is left-handed, but when together in a triple helix, it is right-handed. Yes, Gabrielle, that's correct. So make sure to know that. I don't remember if it was tested or not, but I know that RTA last year, he kind of emphasized that. You're welcome. Okay. So know that the triple helix is held together by hydrogen bonds, by the glycines, and then you have your proline groups facing outward because they're too big to fit in the middle, and those are what add, I guess, tighten the triple helix essentially. And that's all I would know from this slide. I've highlighted pretty much everything I just said. Okay, so this is the slide I was talking about. This is the sequence that seems to be pretty repetitive. You see it several times in there. However, you'll also see proline in the second spot as well, and then something else here. I think the most important thing to note about this is that glycine, proline, and hydroxyproline are found frequently in the, strand or the sequence. So this is the primary structure, and then that would help form the secondary structure, which ultimately determines the tertiary structure. Okay, so I think that was the main point here. Okay, for this, the only number I would remember is maybe that glycine makes up 33%. That's pretty obvious because, if it's in every third space, it's going to take up 1/3 of all the amino acids, which is 33%. I think I remembered from last year that alanine was more significant in elastin, but that's lower yield anyway. Elastin is just another type of fibrous protein that actually just adds elasticity to tissues. So we didn't really talk about it that much, but it might be important to note that you can also find almost the same amount of glycine in elastin as well. So it's got to be important for structural proteins. And then also she emphasized that you only see these three really in collagen, and then maybe just see one of these in elastin. So these are pretty specific to collagen. And I think that's the point she was trying to make. So, but don't worry about memorizing any of these numbers. Probably just remember that it's the 33% of glycine. Okay, so she was going to start talking about the striations here, which she goes into in better detail at the end of the lecture. So you'll see that the collagens are lined up from tail to head, and that they will have these intramolecular bonding, which we'll go into in greater detail, and all of this would form a fibril. I think I memorized these numbers, but I think it's really low yield. Maybe just remember that 1,000 amino acids make up a collagen strand. But yeah, don't worry about it if you don't get to it. I don't think she's going to be nitpicky about the details. Okay, so for the structure on these, don't memorize the structure at all. Memorize these names, though. These are super important and very high yield because she will ask you about them. I think it's important to know that these are the hydroxylysine, or the lysine and 5-hydroxylysine, as well as histidine. So this is something you don't really think about because we didn't really go over it, are important for the covalent cross links that hold the fibrils together, as well as the individual strands together. So we'll go into that, but know that histidine is also one of the ones that do the cross linking. Okay, so once again, don't remember the actual like chemical structures, but do remember all of this. Know that it's you need oxygen for this. I memorized alpha ketoglutarate, which is something that is produced in the TCA cycle, and that we'll learn, I think, in next block. It's probably lower yield, and I think she mentioned that it's not that important, but I don't know, I remembered it. Okay. And so this is pretty much the hydroxylation of only the proline. So I'm assuming that since we didn't go over the hydroxylation of the lysine, that it would probably be very similar. But regardless, know the enzyme, know that you need vitamin C or ascorbate, and then know that you need oxygen as well to make the hydroxyproline.
Phoebe, she did go over the lysine amino acid oxidase where the lysine residues are cross-linked. Is that what you were saying?
Oh, no, I mean to hydroxylate the lysine. So like to make 5-hydroxylysine. So yeah, she does go over how to cross-link them, but to make the actual like hydroxyl groups on the lysine, I think it's different, but I don't think she went over it. Does that answer your question?
I think so. Okay.
Oh, we'll go over it and I'll tell you what I'm talking about. Okay, so remember that glycosylation only happens on hydroxylysine. I don't think it happens on lysine because you do need the hydroxyl group that is added onto the lysine in order to make the sugar ester. So know that the sugar that's added is galactose, and know that it's glucose as well. So know those two. Don't worry about this, and don't really worry about the structure. I would just know that you add the sugar group to hydroxylysine and that it helps stabilize the triple helix. Okay, so if there's anything to memorize from this lecture, it is this whole thing. So I'm going to post one of my review sheets, and I kind of took this whole thing, I rewrote it, and then added like the enzymes and the necessary and important information to it, so it's all kind of in one place. And I'll post it later tonight so you guys can see what I'm talking about. But I strongly recommend drawing this out with all the added information and then kind of reciting it over and over again to memorize this pathway, because it's super important. It's also important to know where everything is taking place. So whether it's in the ER and Golgi compartment or outside of the cell, you have to know that. So essentially, you have mRNA that goes through the ribosome, and then that forms your single alpha helix. So then your alpha helix is then going to be hydroxylated on the prolines and the lysines. So you saw the hydroxylation of the prolines already, so I would write down the enzyme responsible for that, and then vitamin C and oxygen to your study sheet. So then after that, then there is glycosylation of the hydroxylysines. So that's only going to be added, the sugar group is only going to be added to the hydroxylysines. Okay, following that, you will have the 3 alpha helices being put together. It's going from the C terminus to the N terminus in terms of how it's winding together. And this is determined by the cysteines in this propeptide, which is ultimately going to be cleaved off. So these cysteines make disulfide bonds, and this kind of gives it its directionality in terms of how it winds together. So once it's done being wound up, it gets secreted. So outside the cell, you're going to see this pro-collagen molecule, which is then going to be cleaved to make your mature collagen molecule. And then it gets assembled into fibrils. And remember, the fibril is held together partially by the covalent linking between the lysines and the hydroxylysines. And then all those fibrils will come together to make a fiber. So don't get these two confused because they sound alike. But yeah, know this forwards and backwards because she's going to have at least 2 questions on it. Okay, so this is just your regular lysine, and it's going to be, or the N group or the nitrogen group is going to be turned into an aldehyde. So it could either, both lysines can either both become aldehyde groups that then react with each other, and then that's how they cross-link, or one of these lysines will have the aldehyde group added, and it just reacts with the regular old lysine, and then it also covalently links through that means. You don't have to know these names. She doesn't want you because that's orgo, and it's not important. But just understand that it could either be turned into an aldehyde and then react with a regular lysine, or it could be 2 aldehydes that react together. But definitely know this enzyme. That's super important because she's definitely going to ask you about that. So these don't necessarily have to be hydroxylysines, but they can be. So I think this is what you're asking about, that they didn't show us how to make a hydroxylysine. They only showed you how to make a hydroxyproline, but here they're just showing you how to covalently link the two. So this could also be a hydroxylysine, or it could just be 2 lysines. It doesn't matter because this is just showing you the covalent linking. Okay. Sorry, Elizabeth, were you going to say something?
Yeah, I think I just clarified that it's the lysine, hydroxylysine, and histidine that are involved in cross-linking. That's basically all it is. We're gathering from that slide.
Right, exactly.
Thank you.
You're welcome. Okay. So through these covalent links, you're going to be forming essentially the fibrils. So these are individual collagen molecules that have these covalent linking within their triple helix, or they can have intermolecular crosslinks that help hold the fibril together by allowing it to associate with neighboring collagen molecules. So this could be a linkage between the hydroxylysine or the lysine, or 2 lysines, or whatever. So know the difference between inter and intramolecular cross-linking, and know the importance of this intermolecular cross-linking in the formation of a fibril. Okay, and know that they overlap by 1/4 of the whole length of the collagen molecule. So I remembered this 1/4 staggered fashion, which helps makes the striations. I would remember this number. It's probably low yield, but I remember remembering it. So I don't know. It might be important. Okay, so this is pretty much just a summary. You have your left-handed collagen helix, and then it gets associated with 2 others and makes a right-handed triple helix. So ultimately, each collagen molecule is going to be set together with overlapping by 1/4, I believe. And then that is what causes these little gaps, which help form the striations in the fibril when you put them all together and kind of zoom out. Yeah, I think that's all this slide is pretty much showing you.
I had a question. I just wanted to clarify that covalent bonds are holding together the fibrils.
Yeah, so it's the covalent bonding between the lysine and hydroxylysine, or the histidine, or whatever, that is kind of letting these link together and, form like these staggered associations. And ultimately, that's what holds the fibril together. Does that answer your question?
Yeah, thanks.
Awesome.
Hi, Phoebe. Can you go into a little bit more detail on the intermolecular and intramolecular crosslink? I think it was, yeah.
Yeah. So I think we went over that where it's the hydroxylysine and the lysine, they, are converted into aldehydes, and then they, they connect. So that's what this is. Does that answer your question? So your intermolecular crosslink is going to be between two collagen molecules, and then your intra is just going to help stabilize the triple helix by itself. So it doesn't really help with the fibril formation as much as the intermolecular crosslinks. Is that what you're asking?
Yes, thank you.
Okay, awesome. Okay, so this table, which is a lot, I will let you know that you don't have to memorize any of these at all, because that's really just saying that type 1 has two alpha helixes of the same type and one of a different type, but don't worry about that. I would maybe memorize 300 nanometers long fibrils. I mean, it's probably low yield, but I remembered it last year. The most important thing to note is that 1, 2, and 3 are the most common types of collagen. And you'll actually have to memorize these when you learn it in histo next semester. So don't worry too much about it. So type 1, I would memorize that this is associated with bone. And when you think of bone, you think of ligaments. Ligaments are kind of similar to tendons, I guess. So I associate these 3 together. And then dentin, which is like a part of your tooth formation. And, teeth, bone, they're all associated together. The things I would probably have to memorize that's not linked to all this is these 2, because I don't really associate those with bone. But the way to remember this is the word bone has the word 1 in it. So you can remember that that's type 1 collagen. And that's probably the most important one to remember. So type 2, remember that type 2 and type 9 are kind of associated with each other. So they're both going to do cartilage and the vitreous humor. So the vitreous humor is actually just the jelly substance that makes up your eyeball. So in case you didn't know that, that's, what it is. So I would memorize that these 2, cartilage and vitreous humor, are for 2 and 9. And then type 4, 4 is all your basal laminae. So what your basal laminae is essentially it's your basement membrane. So the basement membrane is this structure that helps essentially anchor the epithelium onto the tissue below it. So I'll show you a picture in the next slide. I kind of drew out what I'm trying to say. So because it's the basement or the basal laminae, basement, you think of the floor. So floor, because it rhymes. So yeah, that's how I would remember this. So I would definitely remember the representative tissues and then these. So don't worry about this one. That one's low yield also. But just remember the ones I highlighted.
Pardon me, Phoebe.
Yes.
I have a question, and Pierce, please correct me if I'm also wrong. I wrote in my notes that 80 to 90% of all collagen is part of type 1, type 4, and type 9, not type 1, 2, and 3, which you have on your slides.
Did she say that in the lecture? Because you might be right.
Yeah, she did say that in lecture. She said 80 to 90% of all collagen is type 1, type 4, and type 9. Pierce, correct me if I'm wrong.
Okay, I might have just heard her wrong, so you're probably right. I'll double check for you.
I
I have
have in
type
my
3.
notes that it's 1, 2, and 3 for 85 to 90% of the total collagen.
I also have 1, 2, and 3.
Yeah, me too. Okay, thank you guys.
Okay, cool. Alrighty. So yeah, it's probably not going to be tested like which are the most. Yeah, no, there she's not going to ask that, so don't worry about it too much. I would definitely remember the representative tissues though. And then she does talk here a bit more about the association between type 2 and type 9. So know that they're kind of like linked together, and then they ultimately help form the structure of cartilage together, and it just adds strength. You don't have to know the details of this. It's just associate them together, and that's why they're found in the same tissues as each other. And then she just wanted to show you a picture of type 1, which is, the one found in bone and whatnot. Okay, so this is what I was talking about type 4. So you have your epithelial cell, below it is your basement membrane, and then below that is your connective tissue. And then ultimately the basement membrane has these little proteins that kind of anchor the epithelial cell onto the connective tissue. So you'll find it in most epithelial cells. So what it forms is this kind of like mesh-like structure with all these proteins going through it, and then ultimately your cell would be found above it or whatever. Really, you don't really have to know too much about this. Maybe associate laminin with it, but it's just kind of showing you that it forms like a mesh-like network to kind of connect things. Did somebody have a question? No, okay. I highlighted these just to know like the words for these, but she's probably not going to ask you that. I think she just wanted to show you just these like examples of what type 4 collagen can form to make this whole mesh-like thing. Okay, so for the diseases, she said not to worry about them too much, but I remember memorizing them, and I'm not sure if it was on a practice question from the TAs last year or if it was actually on the test, but I do remember actually answering a question about it. So just to be safe, I'm going to kind of go over it a little bit. What I would know is that to associate these with collagen issues. So, you can have all kinds of issues with collagen formation due to like a genetic abnormality or a post-translational modification, or, if you have like low vitamin C, for example. So, osteogenesis imperfecta is just, a inherited condition that causes brittle bones. So you know that it's going to be type 1 collagen because remember, type 1 is what makes up your bones. And then when it gets, you can have it in the recessive or the dominant. And obviously, if it's dominant, you're going to have a more severe form of it. I just remembered the highlighted terms, so don't worry too much about the details. For this, it's important to know that Ehlers-Danlos allows for joint hypermobility. You'll easily be bruised, and then your skin's more stretchable. And this is lower yield, so don't worry too much about that either. This is probably what she'll test on, I think. Know that these symptoms are associated with scurvy, and then know the why. So remember that when you're hydroxylating lysine and proline, you're going to need ascorbate, which is vitamin C. And we know that scurvy is what, pirates get from spending too much time at sea, and they don't get their vitamin C. So, yeah. All right, so this, I just remembered tissue collagenases and Clostridium collagenase. So tissue collagenases are these enzymes that will break down your collagen. And sometimes these are used when you're trying to heal a wound, but other times they're used for by tumors. So your tumor cells will secrete these, and they'll like break down the basement membrane so your tumor can go deeper into the subcutaneous tissue and invade your blood vessels and all that, and then spread. And then Clostridium, this is a collagenase. So this is toxin O, is a type of collagenase that is secreted by Clostridium, and then that leads to gas gangrene. I would just associate these together, and then maybe understand that COPD is associated with collagenases due to the breakdown of your alveoli in your lung, and then as well as cancer metastasis, which I just described. This is just pictures, not important. And then she goes into the summary. Okay, so definitely answer these questions. So yeah, does anyone have any questions about that? Did I miss anything in the chat?
Can you please go over the tissue collagenasis and the Clostridium collagenasis, please?
Yeah, okay. So what did you want to know about this? It's just like an enzyme that breaks down collagen, right? So when you have a tumor, the way it metastasizes is by secreting these tissue collagenases, which will break down your basement membrane or your basal lamina, for example, and then that's what allows it to invade deeper in the tissue. Because remember, the basement membrane is what holsters the cells in the epithelium away from the subcutaneous tissue where you'll find all the blood vessels. And then for the Clostridium is just, I think, a bacteria that, its toxin that it secretes is toxin O, which is a collagenase. And then that leads to gas gangrene. You don't have to know the details about it, really. It's just association of these words. Does that answer your question?
Yes, it does. Thank you.
Awesome. Great. Okay. So before we get to the next one, let's take a 3-minute break, and then we'll get back here at 9:39. Does that work for everybody? You're welcome to ask any questions on this lecture. Okay, great.
Hi, Phoebe. I'm a little bit confused still on the role of histidine in collagen.
Okay, so it's just a minor detail, and it's pretty much the same thing as, so here. These covalent cross-linkings that happen between lysines, and they ultimately form these intermolecular cross-linking that holds the fibrils together. I would just know that these linkages can also occur between like lysine and histidine. It's a super minor detail, but she didn't really go over it, so she likely won't test on it, but it's good to know. Does that answer your question?
Yeah, thank you.
Awesome.
And, for the intermolecular ones, is it specific to lysine and lysine, whereas the intramolecular is specific to hydroxylysine and lysine, or does it matter?
No, it doesn't matter. Sorry, I just drew that as like an example, but no, it doesn't matter. It could be between any of the above.
Okay, thank you.
You're welcome.
And is the glycosylation also to stabilize the intermolecular crosslinks, or is that just for the triple helix?
Yeah, so glycosylation is really just for like stabilizing the triple helix. So it doesn't necessarily play a role in like the intermolecular cross-linking or anything like that.
So it would probably just be intramolecular then?
For the glycosylation?
Yes.
I don't think it would be classified under intramolecular, but like I guess intramolecular, associations, because it really doesn't bond to anything. So I think just like the largeness of this sugar group maybe does the same thing as like the proline chain, where it kind of allows it to tighten because of the steric hindrance.
Okay, thank you. That makes sense.
You're welcome.
Phoebe, can you go back to the slide with the diagram that we should know?
Oh yeah, it looks like a dolphin. Yeah, this one.
Yeah, can you explain what part of the cell like each section is?
Yeah, of course. So so this whole white area, this is the ER in Golgi. So all of this, so up to the formation of the triple helix, is made in the ER in Golgi, right? Then after that, it's secreted. So nothing happens in the secretory vesicle. It just gets secreted. And so all of this and that is outside of the cell. So,
sorry, I can't see your pointer.
Oh, really? Okay, sorry.
Oh, it's okay.
Better?
Yeah.
Okay, awesome. Okay, so what I was saying is that all of this in this white bubble is what's happening in the ER and the Golgi compartment. So know everything up until the formation of the triple helix happens in the ER and Golgi. Then it gets secreted and it goes into the secretory vesicle, but nothing happens in the secretory vesicle. It's not until it's actually, secreted outside of the cell. So this is all outside of the cell that it, cleaves the propeptides, forms a mature collagen molecule, then assembles into fibrils and ultimately to fibers. Does that answer your question?
Yes, thank you.
You're
And
welcome.
now those cross-linking with the lysoamino oxidase, is that going to be outside of the ER? Is that like a post-translational modification outside of the ER? Is that also like in step 4 or something?
So I'm assuming that it will probably be helping in the assembly of the triple helix for the intramolecular bonds. And then it isn't until it's also used for the formation of these fibrils. So it will happen technically in both.
Okay,
Hi,
thank
I have a question about that also.
Yeah.
So is the fibril referring to like just a bunch of the triple helixes together?
Yeah, it's a bunch of triple helixes together, and then they form a fibril, which ultimately come together into a fiber.
Okay, and are they connected by covalent bonds or hydrogen? The fibril, is it held together like in any specific way?
So the thing that we learned is that it's held together by these covalent bonds.
Okay.
So whether there's like some other kind of associations forming, I'm not sure about those, but I know for a fact that they're held together by those covalent bonds.
And then is the triple helix, so that's held together by covalent bonds, or is that still hydrogen bonds?
So the triple helix is mostly held together by the hydrogen bonds between the glycine residues, but then you also can have the covalent bonding within it as well. So kind of both.
Thank you.
You're welcome.
It's Phoebe. So the cross-linking it also occurs in the collagen fibril, but in the collagen fibril, it's held together by covalent bonds.
Right, yeah, within the collagen fibril, you'll have covalent bonding, yes.
Okay, thanks.
You're welcome.
Can you just explain again the first part and where you have like ribosome RNA to DNA?
Yeah, okay, so you have your DNA that's forming an mRNA. This is all somewhere else, so the DNA is within the nucleus, etc. So you know that your rough endoplasmic reticulum is covered with these ribosomes, right? So these ribosomes are going to be hanging out here. They'll pick up some mRNA, and then the ribosomes will pump out the polypeptide chain, which in this case is going to form an alpha helix as soon as it gets out of the ribosome. Does that answer your question?
Yes, thank you so much.
You're welcome. Any other questions about this? Okay.
Phoebe?
Oh, yeah.
Sorry. Can you
It's
go
okay.
over the hydroxylation of the proline residues again and like what the effect of that is?
Okay, so the hydroxylation of the proline residue. So they're really just there to add stability. It's the hydroxylation of the lysine group, so the hydroxylysines, that allow for the sugar group to be added, which also adds stability. So hydroxylation, for the most part, is just adding stability to the whole polypeptide.
Okay, thank you.
You're welcome. Okay, I'm guessing nobody has any other questions. Okay, so I guess we'll start the next lecture. Okay, so this one's a little bit harder in my opinion, but once you go over enzyme kinetics, which last year we had right after this lecture, but for whatever reason I guess they moved it towards the end of this block. It'll help a little bit with understanding this a little bit better. So the main idea of this slide is she was just showing you the movement of hemoglobin. So your lungs have a high amount of oxygen in them. Your hemoglobin picks up the oxygen at your lungs, goes to your tissues, and releases the oxygen, in which case myoglobin kind of snatches up this oxygen and kind of stores it. So that's really all you have to know of this slide. It was just like an introduction to that. So for this, she was kind of showing you that all of these have a common ancestor that they evolved from. You've got your alpha hemoglobin, which is just your alpha chain, and then your beta hemoglobin, which is your beta chain. Remember that your hemoglobin is a tetramer, and it's made up of 2 alpha and 2 beta subunits together. And then your myoglobin is a monomer, so it's, I think, analogous to your alpha hemoglobin chain. Okay, so this is her showing the same thing. Again, you have sequences between alpha hemoglobin and myoglobin that are identical, and then you have the yellow highlighted ones that are conserved, meaning that even though they're different amino acids, for example, in valine and leucine, they're both similar because valine and leucine are both hydrophobic and they have small side chains. So they don't really alter the tertiary structure that much because they're so similar. Okay. So this is just showing you once again that the primary structure is a little bit different, but because of all the similarities, it allows for the tertiary structure to look very similar between alpha hemoglobin and myoglobin, which is a monomer, and then this is the alpha chain, so one subunit of the hemoglobin. And then know that globins are essentially just soluble metalloproteins, so these are proteins with a metal group in it, in which, in this case, iron within the hemoglobin, or the heme group within the hemoglobin. So you've got your hemoglobin, you've got your heme group within it, and then the iron within the heme group. Okay, so here she was just kind of showing you the difference between the myoglobin and the hemoglobin. So we know your hemoglobin is going to be a tetramer. So you've got 4 of these, and that your myoglobin is a monomer. So it's just going to be one protein. So it's going to look more similar to your alpha subunit on your hemoglobin. So you've got 2 alpha subunits, which are identical to each other. So these 2 are identical to each other. And then the 2 beta subunits are identical to each other. Although between the beta subunit and the alpha subunit, they're similar but not identical. And I think that's the main point she wanted you to know in this slide. Oh, and you also want to know that the myoglobin and the hemoglobin are both predominantly made up of alpha helices, similar to collagen. However, it's a globular protein and not like a fibrillar protein.
Sorry, Phoebe, what did you say? You said that the beta were not identical?
Okay, so the two yellow subunits, these 2 are the beta subunits. They're identical to each other. However, the alpha subunit and the beta subunit they're similar, but they're not exactly the same. They're not identical.
Okay, thank you.
You're welcome. Okay, so this is just kind of showing you the same thing, but she drew it out a little bit differently so you could kind of see better. You've got your protein, your alpha helices here, and then you've got your heme group in the middle, and same here. So your hemoglobin technically has 4 heme groups because it has 4 of these proteins put together into a quaternary structure, whereas this just has a tertiary structure. Okay. So you don't have to remember this structure at all. I would just know the word chiral ring and know that it is there are 4 of them, and they're each linked to the iron group. So at this point, your iron still has a 2+ charge. So it has 2 groups that it can interact with as well. Because iron is a tetrahedral and therefore has 6 iron ligands total. And know that there are 4 chiral rings that associate with the iron are known as a tetrapyral. And that they all lie within the same plane. for the most part, except for, when we learn later that the iron kind of puckers out a little bit. So know that this heme group is the functional part of the hemoglobin or the myoglobin. And I think that's all you have to know. So don't worry about the structure too much. Just associate pyrrole ring is one of these rings. 4 of those are called a tetrapyrrole, and they're all associated with the iron group. Okay, so here she was kind of showing you if you're looking at it from a different angle. So you've got your pyrrole groups in these gray dots here. And then you've got your proximal histidine, which is coming off of this alpha chain. And then you've got your distal histidine, which is coming off of this alpha chain. And remember, this is all one subunit of hemoglobin. So it's not like multiple subunits interacting with each other just yet. So you've got your proximal histidine, which actually can bind to the iron. So this is your iron, and it's going to bind to it. And then your distal histidine, which is a little bit too far to actually interact with the iron, but it's important for another reason, which we'll get into in the next slide. And I think that's all she was trying to let you know with this slide. I think that's it.
Phoebe, are you gonna post those like study sheets you made for these 2 PowerPoints?
Yeah, so I'm still working on the one for this lecture, but I'm gonna probably be done with it by the end of the day. So I'll post them, tonight.
Okay, thank you.
You're welcome. Okay, so here you're looking at one subunit of hemoglobin, and it's kind of showing you a better picture of these histidines. So your distal histidine is just a little too far, so it's not binding to the iron, but the proximal histidine is. So the purpose of this distal histidine is it kind of forms this interaction and kind of makes like the perfect pocket for oxygen to bind into. So it's important for like the structural component of the heme group, or like the surrounding protein. So it makes it like a pocket for the oxygen to bind more easily. So I think that's all she was trying to convey in this slide. Okay, so this is where it gets a little, yeah.
So oxygen binds to the distal histidine?
No, it doesn't bind to the distal histidine. So what she was trying to show you is that this distal histidine kind of makes a kind of like a pocket that allows for the oxygen to come in here and bind to the iron. So the oxygen binds to the iron, yeah. And the distal histidine just kind of makes the perfect little environment for it to bind.
So the distal histidine creates the site, or helps create the site for oxygen to bind.
Exactly, yes.
Okay, thank you.
You're welcome. Okay, so myoglobin, remember that myoglobin is found in your tissues. So your tissues will always have a low oxygen concentration because they're far from your lungs, and your lungs will have a high oxygen concentration because that's where you get all your oxygen. So because of this, myoglobin has to be really good at snatching up any oxygen it can find, which is why it will form a hyperbolic curve. So you'll have just a teeny tiny increase in oxygen, and it's already picking up 50% of it, right? Or it's saturated 50%. So remember that one myoglobin has 4 binding sites for oxygen one binding site for oxygen. So it'll be able to pick up 50% or saturate itself 50%, hypothetically. And then if you just increase it a little bit more to about 25 oxygen concentration, remember partial pressure of oxygen is analogous to the concentration of oxygen, it'll be able to pick it up almost like 98% or something like that. So it's not really specific to the oxygen concentration because it's just a really good grabber. And that's what she was trying to convey with this, graph, is just that myoglobin will show a hyperbolic curve because it's not really dependent on the oxygen concentration the same way that hemoglobin is. So,
excuse me, did you say where the myoglobin, if it's picking it up, does it just hold on to it, or does it convert or take it anywhere?
I think what it does is it just stores it until the tissue needs it. So like if it's in a muscle, you know, when you start working your muscles and using more of the oxygen and metabolism, it's going to start like releasing it to, you know, be used.
Okay, thanks.
Yeah, so myoglobin is really just a storage protein for oxygen in the tissues. Okay. In contrast, you've got your hemoglobin. So remember, hemoglobin is kind of a transport protein. So it kind of has to have an idea as to where in the body it is, because you don't want hemoglobin to just start releasing oxygen as soon as it leaves the lungs, because then it's not going to work very good. So in order for it to know where it is in the body, it has to kind of interact in this kind of a sigmoidal curve type thing, where it is a really good grabber of oxygen when the oxygen concentration is high, like in your lungs, and it's really good at releasing oxygen when the oxygen concentration is low, like in your tissues. So hemoglobin is more so dependent on the concentration of oxygen than is myoglobin, and that's why it creates this sigmoidal curve, where if you have low oxygen concentration, you're going to have a low affinity for oxygen, whereas at a higher concentration, you're going to have an increasing affinity for oxygen. And yeah, okay. So here she was showing you what would happen if hemoglobin didn't have this cooperativity. So if you didn't have cooperativity at a lower concentration, it'd be a, I guess, better releaser of oxygen, just slightly. The difference is maybe like, I don't know, like 0.05 or something like that. And then if you had, if you were at the lungs, it wouldn't be as efficient at grabbing the oxygen. So you have this huge gap between normal hemoglobin and less cooperative tetrameric protein that's analogous to hemoglobin, hypothetically, that would have less oxygen bound to the hemoglobin, and then when it gets to the tissues, it's probably only going to be able to release the 2 oxygen that it picked up from the lungs. So essentially, it is going to be less efficient. And that's why you need cooperativity, which essentially tells the hemoglobin where it is in the body.
So this was, she was just showing us the in that graph, in that picture, she was just showing this no cooperativity is also a tetramer, just it doesn't have that function.
Right, yes, it's also a tetramer, this hypothetical protein. It just doesn't have the cooperativity between the subunits, meaning that the subunits each have the same affinity for oxygen and they don't depend on each other to determine the affinity for the next subunit. Does that answer your question?
Yeah, thank you.
You're welcome. Okay, so this slide will make a lot more sense when we learn about the enzyme kinetics. I think somebody's mic is on, I'm not sure. Oh well. Okay, so the idea she was trying to show here is if you had this tetrameric protein with 4 binding sites, hypothetically, you would then bind your first ligand, and then it would cause a conformational change in the subunit next to it, which increases the affinity of that subunit for the ligand, hence allowing the next ligand to bind easier, and then, you know, that does the same thing, and it's kind of a chain reaction. So these Ks are the binding constant, and the binding constant is just really a number that indicates the strength of the binding. So as you move along and you have more ligands bound, you'll see that this strength will increase, hence why the binding constant will increase in number. But that's not super important for you to know. It's just kind of like this concept that she was just trying to, you know, give you a clearer picture as to how this works. I also kind of wrote here the difference between the taut form, which is the deoxygenated form in hemoglobin, and then the relaxed form, which is the oxygenated form. So probably know that. I don't know if she's going to use that terminology on the test, but just in case, it might be helpful to know that. And then you know that this cooperativity is also kind of known as allosteric behavior, which we'll get into more when we talk about enzyme kinetics, so I wouldn't worry about that too much. However, when we do learn about enzyme kinetics, it might be helpful to go back to this lecture and read through it again so you have a stronger understanding of it, and you could see it through like a different, you know, perspective.
Phoebe, real quick question. This might not be like related. I always thought allostery, I always associated it with a separate binding site. Am I thinking of something different?
No, it's probably the same thing. I think the idea here is that since it is a separate binding site that binds, so if you have one ligand that binds and then the separate ligand binds here, it kind of changes the ability of the other binding site to bind, and it's just, yeah, multiple binding sites. And we'll get into that when we talk about enzyme kinetics. So
okay, I think I see what you're saying now.
Yeah, sorry, I'm like kind of bad at explaining
No,
things.
I understand. Thank you so much.
Okay. Okay, so here she's kind of just summarizing the same thing over again, where, you know, if you have no cooperativity, you're not as good at picking up oxygen as you would if you were hemoglobin, which has a really good amount, it's really good at picking up oxygen when the oxygen concentration is high. And then with myoglobin, which is really good at picking up oxygen regardless of the concentration of oxygen. And that's why myoglobin is a better grabber, because it holds on to oxygen more tightly compared to hemoglobin when you're comparing it to different oxygen concentrations. I think here she kind of, like, I highlighted this to know that it's like 1.7 times as much O2, just to know that, you know, relationship there. But I don't think it's really high yield to know 1.7 times. But you can memorize it if you'd like to. I don't remember if Ferreira is really nitpicky about the details or not. So I don't know. I'd memorize it just to be safe.
Phoebe, for the charts, would you say we would need to understand the interpretation? Like, would she be, on the exam, would she put like the chart and we would have to interpret?
So I don't want to say that there's no pictures on biochem, because I feel like my TA said that, and then we had a picture on the test, and I don't remember what test it was. So I would, if I were you, I would at least try to understand this idea here. I mean, you have the saturation of the enzyme, so that's the amount of oxygen that is bound to it, and that's going to increase as you increase the oxygen concentration. I think it's just enough to understand how hemoglobin works in general. So higher oxygen concentration, so at your lungs, which are at 100 partial pressure of oxygen, versus your tissues, which are 20, which has a smaller saturation. So I think that would be enough to understand for this. But I think if she did put a chart or a graph in the test, it would probably be when we learn about enzyme kinetics. So I'm thinking this would probably be lower yield.
Okay, thank you.
You're welcome. Okay. So here she wanted to show you that because the iron is kind of big, it doesn't quite fit in the gap between the porphyrin rings. So when oxygen is not bound, it's going to be kind of puckering down and out towards the proximal histidine, which is also bound to it. So you know here that this is where oxygen would bind if oxygen were present. So when you add oxygen, it's going to bind to the iron and kind of pull it up this way, and then that's what changes the conformation through a series of events to change the conformation of the neighboring subunit. So know that the iron is planar, or it's, you know, within the plane of the porphyrin rings when oxygen is bound, and it's, you know, slightly puckered out when oxygen is not bound. So I think that's enough to know. You don't have to memorize that number. That's too much. Okay, so here she's showing that. So when you have oxygen bound, it moves it up slightly. So here's your iron, which is now in plane with the porphyrin rings. And that's going to pull up the histidine, which is also going to pull at the alpha helix that it's attached to. And that alpha helix is going to be pulled in a slightly this way direction. So this is demonstrating this region between the alpha subunit and a beta subunit, right? So potentially alpha 1, this is alpha 1, and this is beta 1. And then that is going to kind of move things over in the other subunit because they're close together. And if this moves, it's going to move something over here. And that's what allows the neighbor to bind to oxygen easier. So I kind of listed that out right here for you to remember. But for the most part, this isn't super important. It's just kind of showing you the why behind the what. Okay, so this is a summary slide, essentially. She's just saying that at your lungs, you have a higher concentration of oxygen, so you have a higher amount of oxygen bound to hemoglobin, whereas at your tissues, you have a lower concentration of oxygen, so you have a lower amount of oxygen bound to your hemoglobin. And then your myoglobin will be more saturated than the hemoglobin at the same low oxygen concentration. So at 20, for example, it's going to be like 85% because it's a really good grabber. And then she mentioned that myoglobin is essentially the oxygen acceptor, and then hemoglobin is the oxygen donor. And I don't know if she's going to use that terminology, but I wrote it down just in case. Okay, so the Hill equation. You don't have to memorize this equation. I think it's enough to kind of understand what these mean. We know it's a linear equation because if you remember from like math in middle school, it's y equals mx plus b, right? So you know that this is going to be the slope, so the Hill constant. This is the most important thing of this equation and what she will test on. I kind of gave you an idea as to what these variables kind of stand for, but they're really low yield, and she's not going to ask you like what the y means. She might ask you like what this means, but that's more important for like interpreting the graph. So essentially what the Hill equation is, it is used to find the degree of oxygen binding to the hemoglobin. But since we don't have to solve for it or anything, it's not really important to really know like what each of the variables stand for. So this is the most important part of this, is understanding that because the Hill coefficient represents the slope of this line, and this is the y and this is the x, right? So you know, you'll see this in the x-axis and then this in the y-axis, and then so that means that n or h, because they are the same thing, they stand for the Hill coefficient. You'll see that the slope is going to determine whether it's cooperative or non-cooperative. So this is what you need to remember from this. It's the most important thing to remember, because she'll likely give you a test question and give you like the Hill coefficient. You have to determine whether it's cooperative like hemoglobin or not cooperative like myoglobin. But all of this is really low yield, so you don't have to worry about it. And she most likely will not give you a picture of the graph and make you determine it. But just in case, just know that the slope, if it's greater than 1, it's going to represent cooperativity. Okay, so this is definitely going to be tested on. This is probably more important than some of the other stuff we talked about, is understanding the Bohr effect. This is going to come back when you learn about the lungs in physiology, so it's important to understand this now, so it makes it a lot easier when you learn about it in physiology. So essentially what she's saying is that the pH and the carbon dioxide concentration in the tissues and the region where the area is running through, the area of blood is running through, is going to help the hemoglobin determine whether or not it needs to release some oxygen. So we know that at tissues, especially tissues that are metabolically active. So if you're working out your biceps are going to eventually run out of oxygen, right? So they'll start anaerobic respiration and lead to the increased secretion of lactic acid as a byproduct, not to mention the fact that when you are, increasing your metabolism and going through glycolysis at the beginning, you're going to wind up with an increase in CO2. So when you have a decrease in the pH and an increase in CO2, that's kind of telling your hemoglobin that you are near a tissue that needs a lot of oxygen. So that's going to help it release the oxygen to the tissue. So essentially, what you need to remember is that if the pH drops and even if there is no CO2, it's going to help shift the curve to the right to decrease hemoglobin's affinity for oxygen and allow it to be released more easily. And then same thing, it'll shift even more if it's a combination of these two. So hopefully that makes sense. Oh, and then she'll go into the 2,3-BPG, but just so you know, in case she asks, it also shifts the curve to the right because it kind of forms the same function as carbon dioxide and the increase in protons. Okay, so this is showing you why and how that happens. So we know that in the beta chain, so this is in one of the beta subunits, you'll have a histidine, don't remember this number, and an aspartic acid, and don't remember this number either. So when the pH drops, you'll get histidine gets a positive charge. And so this negative charge next to it is going to kind of stabilize it. And when it's stabilized, it's going to kind of stick in this conformation that prefers deoxygenated hemoglobin. So it's going to make it easier to kick off the oxygen and then prevent oxygen from binding. So it lowers the affinity for oxygen. She's just kind of showing you this, but it's not as important as this interaction here. Okay. So for carbon dioxide, on the contrary, it's going to, well, when it's in high concentrations, it's going to interact with the amino terminus of one of these alpha subunits. And that's going to create this carbamate, which might be important to remember and know that carbamate has a negative charge. So this negative charge is now going to interact with neighboring positive charges. And that's going to change the structure of the hemoglobin to favor a deoxygenated state. So similar to the protons, it's just going to stabilize it and prefer the deoxygenated state so that it makes it easier to release oxygen and harder to pick it back up. Okay, and lastly, she goes over this 2,3-biphosphoglycerate. It's just something that floats around in your blood, and it's got a -5 charge to it. I think somebody's mic is on. Okay. So it's going to interact with positively charged amino acids between the two beta chains. So it's really just working between two beta chains, even though it's found in the middle of all four. It's going to interact with these positively charged. I mean, don't worry about memorizing which ones they are because that's not super important. But just understand that they're going to be the positively charged or basic amino acids. And then that helps stabilize the deoxygenated structure as well. So it's kind of the same idea where it doesn't want to pick up oxygen and makes oxygen more easy to kick off. And this is showing the exact same thing. It's good to know that you have 8 positively charged regions on these amino acids, and they are stabilized by the 5 negative charges on the 2,3-BPG. But it's pretty much just showing you a different picture of the same thing that we just went over. As for this, she said not to memorize them, but she didn't say that in my year, so maybe you guys are just not going to get tested on it for whatever reason. At most, I would probably remember this one because it's the most common. So if you have an abnormal solubility of your hemoglobin, it's not going to work properly, and it's probably going to affect the functionality of the red blood cells. So because your spleen is what's going to filter out your red blood cells in your body, so it filters out old and damaged red blood cells, it's going to create splenomegaly, and then you're also going to have mild anemia because more of your red blood cells are going to be destroyed rather than created. So with this, you have a decrease in oxygen affinity of your hemoglobin for whatever reason. In this case, it's this, so don't worry about memorizing these. But obviously, if you can't carry enough oxygen to your tissues, you're going to wind up with cyanosis, which is just, blue skin, essentially. And lastly, we have this, which is you have a ferric heme instead of a ferrous, which is the normal heme. So this is also going to alter your hemoglobin's ability to carry oxygen and ultimately lead to cyanosis as well. But this is lower yield, so I mean, remember if you have extra time, but don't worry about it too much if you don't. And then the summary and the self-assessment, you should definitely do that. So does anyone have any questions before we do this Kahoot? Hopefully it works.
Can you please go over the slide? It was like a couple slides back. The deoxygen, like with the 2,3 BPG. It was before this one.
This one?
No, the one. Yeah, this one.
Okay. What were you confused about?
Basically, the BPG is just supposed to stabilize the oxygenated hemoglobin. Is that like?
So it stabilizes deoxygenated hemoglobin. So it kind of does the same thing that the carbon dioxide and the protons do, but in a different way. So it's going to exist at the interface, so between all 4 subunits, but it only interacts with the beta subunits. So its negative charges are going to stabilize the positive charges of the 2 beta subunits at the same time, and allows for these subunits to kind of conform in such a way that they prefer to kick off oxygen. Does that answer your question?
Yes. And another slide before this, I believe.
This one?
No, before that one, sorry. I don't know the numbers.
Yeah, it's all right, me neither.
Yes, can you explain this one more time?
Yeah. So this is how the pH, so hydrogen ions specifically, work to prefer the deoxygenated conformation of a subunit. So this is your one of your beta subunits. So it could either be beta 1 or beta 2. Remember that your hemoglobin has 2 beta subunits. So in these beta subunits, you have this aspartic acid and then the histidine, right? So your histidine, when it hits below a certain pH, so if you have an increase in a proton concentration, you're going to have a drop in the pH, and these protons are going to be able to protonate the histidine, and that gives it this positive charge at a certain pH. So this positive charge is going to interact with the aspartic acid and kind of stabilize it so that it is now going to prefer the conformation that prefers the deoxygenated state of hemoglobin. So you're going to have a smaller O2 affinity, and so you're going to release more oxygen and not pick up as well. Did that answer your question?
Yes, thank you, Phoebe.
You're welcome.
Can you also go over the carbamate one more time?
Yeah. Okay.
Never mind.
No, go ahead.
I was just going to say before we leave the slide really quick, if the aspartic acid and the histidine are both now more positive, why does that work against binding a negative oxygen?
So remember that the aspartic acid and the histidine will have different pK values. So the change in pH for each required to change the charge would be different. So while a drop in the pH will change the charge of histidine, it may not affect the aspartic acid as much. Does that answer your question?
Yeah, thanks.
Awesome.
Maybe on that same slide, sorry, before you go.
Yeah, it's okay.
So every time that you have like 2,3-BPG or the decrease in pH or increased carbon dioxide, it's always stabilized by the beta, what's it called, monomer? The beta strand?
Okay. So for 2,3-BPG and for the pH, it's the beta subunit. But specifically for the carbon dioxide, where it turns into carbamate, this occurs within the alpha subunit. But the alpha subunit can also, this carbamate, which exists on the alpha subunit, can also stabilize the beta subunit. So it kind of works with all of them, although it occurs only on the alpha.
Thank you.
You're welcome. Does anyone else have any questions? Okay. So I guess
Actually,
we could.
wait, I
Yeah.
got one more. Actually, sorry.
Yeah, go for it.
So essentially, with the Bohr effect, is that when the CO2 is increased, that releases the O2. Is that like occurring in tissues, or I'm kind of struggling with figuring that.
So for 2,3-BPG?
No, with the Bohr effect in general.
Oh, in general. Okay, so you know how in your tissue, so when you're away from your lungs, you're going to have more CO2, right? Because your tissues are, metabolizing and they pump out CO2 as a byproduct. So now when you're moving away from your lungs, you're going to have a higher concentration of carbon dioxide. And so because the concentration of the carbon dioxide increases, it's going to be more likely to interact with the hemoglobin in the blood. And same thing with the pH. So if you are undergoing like anaerobic respiration, you're going to be making lactic acid, and then you're going to be pumping out protons into your blood in the tissues in which you're working out. Like if you're working out your biceps, so the blood by your biceps is going to be very highly concentrated in protons. So that's going to make the protons more likely to bump into the hemoglobin and affect it in that way. Does that answer your question?
Sure. Okay. And then the 2,3-BPG stabilizes that deoxygenated hemoglobin. That's what that has the CO2, so it's stabilized with the lesser concentration of
O2. Wait, say that again.
So the 2,3-BPG stabilizes the deoxygenated hemoglobin because there's more CO2 binding, and there's more there's an increase in CO2 concentration, and the O2 is being released as it keeps going further away.
I think these are like all separate processes. So the 2,3-BPG is just something that is found in your blood, and when you take the hemoglobin out of your blood, it will function better than it would in your blood because of the existence of 2,3-BPG.
Okay, thanks.
You're welcome. Okay. Yeah. Yeah.
Back at the PowerPoint. In the original structure of in the first few slides that she shows the protoporphyrin, those chiral rings are those I don't know, it's hard for me. I know the side chain group, but it's hard for me to see the histidine ring here. The histine ring here.
So which slide is that? One of the first ones?
The first ones where she shows the structure of heme.
Okay, I'm gonna go find it. This one?
Yeah.
Okay, so this is looking at it directly, right? So the oxygen would be pointing out towards you if you're looking at your screen, whereas I think the proximal histidine would be pointing away towards inside of the screen, so behind the iron group. Is that what you're referring to?
Okay, that's a histidine group, or that was what I was confused about. Is it? I don't even. I'll look at it later. Thank you. I appreciate it.
Okay. Any other questions? If not, we can try to start this Kahoot. So.
Can you put the link to the Kahoot in the chat?
Yeah, so for Kahoots, the way it works is you just go to kahoot.com, and then at the top corner, you'll press play. And then when I open this link, it's going to share like a code for you to use once you press play. So it's not the same link. Does that make sense? Here, I'm going to try to.
Yeah, I see it, Phoebe. Just waiting on the game pen.
Okay, sorry, I'm still trying to open up this link. And all right, did it work for everybody? I hope it did, because let me know if you're still having issues trying to open up the, the website or whatever, if you're still confused about that. I'm going to try to share my screen. Okay, hopefully everybody can see it. Okay, can everybody see it?
Where's game pen? Oh, there it is.
Okay. Okay, so just put in the pin and then put your name and great. By the way, everybody, if you don't mind typing in your name into the chat box before you leave, we're supposed to like kind of have an idea as to how many people come into these review sessions. And I think that's the best way to take attendance. So if you guys don't mind doing that for me, that'd be great.
Do you have to report the attendance every time, or is this like an end of the semester kind of thing?
I think I'm supposed to like just keep track of like the attendance, and then I like share it at the end of the semester. But it's really just, it doesn't affect you guys at all. So if you don't come to these, it doesn't matter. It's just kind of for their statistics to see like how many people are, going to these, if it's worth it to still have TAs and stuff for like the next year. It's that sort of thing. So don't worry about it if you're like, worried that it's going to affect you in some way.
Oh, that's interesting, because I don't know any of the other TAs have asked that.
Really?
Yeah.
We're supposed to take attendance, but like for whatever reason, I can't like download the participants list on this. So maybe they figured out a way to do it, but it just doesn't work on my computer for whatever reason.
Do you have to have exact names? Because it says there's 126 people in the meeting. So I don't know if that makes it easier just as a number.
I don't know. I honestly have no idea. They just told us to download the participants list, but I honestly couldn't figure out how to do it. So I'm assuming it would be names. So I honestly don't know.
Hey Phoebe, I have a question.
Yeah.
So I'm like I want to go type my name in the chat, and it says only owners can send messages to this channel. So I know it's definitely I'm doing something wrong, but can I just email you after class?
Yeah, that works. It's okay. You're probably in here as a guest for whatever reason. I honestly had that problem last year and I couldn't figure out how to change it. So try logging into Teams through your school email and maybe that'll work. Or maybe you just never received like the invite and you're using like an external link. I have no idea.
Yeah, I have no idea. This is like the only one that it says that for is like the TA sessions. Like for class and everything, it's fine. It's just the TA sessions. But I'll look into it.
Yeah, so you probably just like signed in as a guest through this link. Okay, we'll start the Kahoot. Sorry guys. Thank you so much for helping me out with that.
How do we check in for attendance again?
Just type it in the chat, just your name.
Okay, thank you.
You're welcome. Thank you guys so much for helping me out with that. All right, we'll start the Kahoot now if you guys don't mind. So hopefully everybody signed in okay.
Are we supposed to see the like question?
Yeah, the question, can you guys see it okay? It's like up here.
Oh, I can't see. Oh, maybe I'm just stupid. I see it now.
Okay.
Yeah, you have to go in. You have to like watch the meetings thing and then answer it on whatever you're like doing.
Wait, I don't understand. Where's the question?
It's on my screen. I'm sharing my screen. It's like, you should see it on the Teams meeting.
Oh, okay. So you have to look at
Yeah,
both.
it's actually easier to use your phone for the submission, your answering submission.
Yeah, it's way easier to use your phone so you don't have to open up 2 browsers. Sorry about that, guys.
Did we already close the first question? Because it just said sorry and I didn't even get a chance to read the question and answer.
Yeah, sorry. It's timed, so I gave you guys 20 seconds for this. But yeah, I'd already closed the first question. But don't worry, I'll post it so you can play it again on your own time. Okay, so anyways, we'll go over this question. So which of the following is not a hydroxylated amino acid in collagen? Yes, the green question or answer option is correct. Remember that it's hydroxylysine, not hydroxyproline for 5-hydroxylysine. So that one doesn't exist. All right, is everyone ready for the next question? Okay. Great, awesome, yes. Remember that the alpha helix by itself, the pro-collagen alpha helix, is a right-handed turn. Oh my gosh, wait. Pro-collagen, oh my gosh, sorry guys, I think I got this wrong. The pro-collagen alpha helix by itself is, oh no, it's false, yes. So pro-collagen alpha helix is a left-handed turn. So yes, this is false. Okay, next question. Okay, great, awesome. Remember that you're only glycosylating the hydroxylysine. So hydroxyproline does not get hydroxylated or glycosylated, sorry. Great, yes. So rickets is not. I always get confused between rickets and scurvy, so I posted this so you guys don't get confused between those two. Great, everybody. Yes. So remember, type 4 is fluorobasolaminoase. Awesome, guys. Yes. They're mostly composed of alpha helix. All of these other ones are wrong because they either, refer to just hemoglobin or just myoglobin. So great job. Awesome. Yes, correct. Remember 2,3-BPG, when it's bound, it's going to decrease the affinity for oxygen, which would shift the curve to the right. Great job, guys. Remember, it is a Hill coefficient greater than 1. So none of these of the other options are greater than 1. So it would lead to this one to be the correct answer. So greater than 1 is cooperativity, equal to 1 is non-cooperativity. Awesome, yeah, remember, myoglobin has a hyperbolic curve, not a sigmoidal curve. Okay, and last question. Great job, guys. So yeah, distal histidine does not bind to the iron. It's the proximal histidine that does. Remember, distal histidine just helps form the pocket that makes it easier for oxygen to bind to the iron. All right, who won? Good job, Darla. That's great. All right. So if anybody has any other questions or wants me to go over the Ferreira questions from last week, you're welcome to stay and ask them. Otherwise, you guys are free to go. Thank you so much for typing your names into the chat and helping me out with that. And hopefully this was helpful.
Oh, by the way, Phoebe, I think if you just go to where it says show participants next to the chat box, it's like a little teams. It has little people. It shows you everybody who's in it.
Yeah, so I was trying to copy and paste that, but it wouldn't let me for whatever reason.
Oh, okay.
Yeah, so I could, I'm supposed to have 3 dots next to the word participants. That would allow me to download the participant list, but for whatever reason, my computer doesn't show it. So.
Oh, okay. Sorry.
Thank you so much, though. Yeah, thank you so much.
Hey Phoebe, I had a question about the carbamate and the Bohr effect. Does it just have the same effect as like the salt bridges, or is it different? I think I missed that when you re-explained it.
So I'm sorry, I could hardly hear you. You said, does it have the same effect as what?
The carbamate and the Bohr effect, does it have the same effect as like the salt bridges, or is it a little bit different?
Okay, I'm sorry, I'm like not hearing one of the words you're saying. It's.
Can you hear me now?
I think a little bit better.
Okay, so I was just asking about the carbamate and the Bohr effect. I was wondering if it had the same effect as like the salt bridges, or is it a little bit different?
Oh, I see what you're asking. So the salt bridges. Kind of, yeah. So you know how the salt bridge would be a positive and a negative charge interacting with each other, so it adds stability. So yeah, that's pretty much what it is doing, is just, you know, adding stability through these like salt bridge bonds type things.
Okay, thank you.
You're welcome.
Phoebe,
Yes.
could you go over the hemoglobin and allosteric regulation, like with the ligands and the taught or released, that kind of stuff?
Yeah, I'll open up my slide to that. That was, okay.
I think it's slide 14 or 15. I can't tell.
Okay, 14 or 15. This slide?
Yeah, that one.
Okay, so this is kind of supposed to, can you see my pointer? I'm not sure.
Yeah, I see your mouse. It's like not red, but.
Oh, okay. I could just slideshow and make it easier. Okay. So what this is showing you is an completely unbound, in this case, hemoglobin. So you've got your 4 subunits, and none of them contain the ligand, which in this case would be oxygen. So the first ligand would bind under, certain conditions, favorable conditions, so high oxygen concentration. And then that binding would allow for a conformational change in the subunit next to it to occur, which increases the affinity of that subunit to bind to the ligand. So then, you have the ligand bind to that next one because it's easier to bind, and then so on and so forth. So what these binding constants are supposed to show is they're kind of showing the strength of the bound ligand to the hemoglobin subunit. So as you go in this direction, as you bind more ligands, the strength of the connection between the ligand and the hemoglobin is going to get stronger. So that's just pretty much what it's indicating, but I wouldn't worry too much about that, as she's just kind of trying to show you this concept. Is that what you wanted to know?
Yeah, that was perfect. Thank you.
You're welcome. All right, guys, if nobody else has any other questions, I'm going to go ahead and leave this meeting, but you're free to email me if you have a question that you think of later on. So bye, guys.